So the next speaker is Sofie Valk from the Max Planck Institute for Human Cognitive and Brain Sciences. And they'll be talking today about systematic organization of cortical thickness, co-alterations in substance abuse disorders.
Yeah, thanks for having me. And what? Oh, okay. Thanks for having me. I'm Sofie, and yeah, I'm also learning about substance use disorders by studying it, and it kind of nicely follows the previous talk. So I have no disclosures, first argument, addiction as a brain disorder. So on the one hand, addiction is related to a desensitization of reward circuits, and it also changes conditioned responses, and it strives to be activity. Moreover, addiction is thought to weaken executive function. Also, and this I added a bit short notice, so there's a typo, but the social environment is also very important to support addiction because it makes it also more a habit if your surroundings also enforce this. Moreover, addiction can also be relevant for the developmental stage you're in it and you get in touch with certain substances. For example, adolescence is a moment where there's heightened brain plasticity and people are also more susceptible to become addicted. And last, genes also play a role. So this is super interesting, which makes addiction kind of like this conundrum of all these different factors and how they interact, and the brain is sort of the centerpiece of this stage. So coming from a more neuropsychiatric perspective, substance use disorders can be considered as the #1 comorbidity. They are highly comorbid with many other neuropsychiatric disorders, and they share with them a system-wide structural brain alterations. So maybe they may even enhance already existing alterations, for example in schizophrenia, by on top of them a substance use disorder. This is more general, and I saw this and I realized, okay, well, it is really relevant, right? Like a lot of people that have substance use disorders had a mental illness, but on the other hand, also people with a mental illness sometimes have a substance use disorders. So these things are very much intertwined, and this is also from a care perspective not so easy to know what to target first. And I'm not a clinician, but I also read that in the Netherlands it's very hard to get like these kind of therapies because you kind of need to go at it from 2 different angles. And then treatment is also very hard because not everybody with co-occurring needs get the treatment they need. So going back to the brain, substance use disorders both have unique and shared patterns. They're highly comorbid with other neuropsychiatric disorders, as I already said, and you see like from alcohol use, there is an overlap, like alcohol has widespread impact on the brain, but there's also overlap with cocaine, and there's a shared pattern in more temporal parietal areas, but also areas coming from the insula and more TPJ regions. So now going backward to other work that my group has done with Mackey Hettwer, who's also here in the audience, is that we show that psychiatric conditions show systematic structural alterations. So as you see here, each of these disorders, and these are enigma average maps of Cohen-D, they have different case control patterns at the local level. And our question was, what if the impact of disorder is shared across disorders, across regions? So it's a more of a disorder-like structural covariance pattern. So as you see here, we then looked at which regions would show similar changes across disorders, and as you see, there is a pattern differentiating more temporal regions, which are similar affected across disorders versus frontal areas, but frontal and temporal areas are differently affected across disorders. So, as I said, regions with similar colors are affected together across disorders. So, the question becomes, to what extent similar coordinated co-alteration effects extend to substance use disorders? This remains to be established. So this is what I set out to do. And yeah, here also kudos to Enigma making this so easily available. And we had data from 2,847 patients and 1,951 controls. And as you see, it is all blue, meaning that it's all reductions of cortical thickness relative to controls. And all these patterns are slightly variable. So alcohol shows very strong reductions overall, but also cannabis, opioid, nicotine, cocaine, and vitamins show these differences. So our question was then, how then the impact of substance use disorder across substance use disorder would be systematically organized. So for this, we did this pathological covariance, and we took the top 20% of nodes to look at organizational features of the impact of substance use disorders. And here, there's different theories that one can use to try to understand how these changes in the network come about. For example, there's a nodal stress theory that says that there's certain hub regions that can highly impact it because they're used more. But there's also the epicenter theory that is more related to spreading patterns. So here is the pattern of degree centrality showing like which regions are mostly impacted across disorders in this covariance map. And you see in like yellow brownish, those are the regions with a strong degree centrality. And this is mainly prefrontal areas, bilateral, and also on the right hemisphere, temporal parietal regions. Second, we looked at epicenter models, and here we compared the connectome with a row-wise correlation to see if regions that are impacted in substance use disorders relate to certain connectivity patterns. So on the functional side, we see widespread patterns that are also a little bit asymmetric, especially in temporal areas in both hemispheres, but also in parietal areas, mostly on the left hemisphere, and frontal areas mostly on the right hemisphere. And what is also interesting that if you look at the subcortex, this also relates to amygdalar and hippocampal connectivity patterns. Then, on the structural side, it's a little bit less clear, again, quite asymmetric, but what is maybe also interesting here is to highlight subcortical areas. And then, what is also like the idea of this study, if you compare this maybe to the epicenters that are observed in neuropsychiatric conditions that are mainly in heteromodal areas, both in functional and structural patterns, you see that there's also quite a marked difference between these substance use disorder covariation patterns versus neuropsychiatric conditions. Second, if you then look at covariance, what is interesting, if you compare substance use conditions co-alterations with normative covariance, you see that overall there is a high similarity, with the exception of more dorsolateral, but also medial orbitofrontal areas. And indeed, comparing this then again to substance use co-alteration and neuropsychiatric conditions, you see that here there is again like this decoupling, so the rules don't apply to this medial orbitofrontal and dorsolateral areas. Then we did diffusion map embedding, and to see what underlying rules may guide these alterations. So in the normative data, you see a clear pattern going from more posterior regions to frontal regions, and on the other hand, a pattern going from inferior to superior areas. And with substance use disorders, we see kind of different patterns. So on the one hand, green areas, mostly immediate orbitofrontal cortex regions, to more superior areas, and a second gradient that is a bit of a mess, but I would describe it a bit from frontal to, again, orbitofrontal parietal areas. And comparing this to the neuropsychiatric gradient again, it is interesting to see that the neuropsychiatric gradient shows this differentiation between posterior temporal versus frontal areas, resembling the covariance gradient 1, versus this more posterior areas towards more superior temporal regions. So how to interpret these covariance axes? So on the one hand, you can see this posterior-anterior pattern that is in humans and macaques, and it has a genetic origin, as a pattern differentiating sensory motor from transmodal areas, also linked to development. On the other hand, this inferior-superior pattern, which is also present in macaques, may relate more to the theory of dual origin, like showing the waves of laminarization from olfactory and hippocampal areas. So moving back, comparing this, we see that the first covariance gradient mainly also relates to alterations that is present in neuropsychiatric conditions, but not so much in substance use disorders. And on the other hand, the second covariance gradient, the one differentiating the olfactory areas for hippocampal areas and the laminarization process, is mainly related to both the psychiatric or transdiagnostic gradient, but also the substance use disorder gradient. And then also, if you then do different types of analysis, you see also how both of these patterns are intertwined and may also relate to different organizational processes and how they spread and how they are co-organized. So the conclusion is that the shared impact of substance use disorders on cortical thickness co-varies in a network-like fashion, both related to nodal stress and epicenters. These patterns mirror structural covariance, in particular the differentiation between inferior/ventral and superior/dorsal regions of the cerebral cortex, as probed by a gradient framework. There were notable differences in the OFC, where we see that the OFC kind of does things differences, mainly also possibly to atrophy patterns that are possibly more random in substance use disorders. We see a marked differentiation between the shared impact of substance use disorders in brain organization versus the impact of neuropsychiatric conditions. So with this, I also ended, and a big thanks to everybody involved, in particular Matthias Kirschner, who helped me with the data.
Are there any questions? We have time for maybe one or two. No, I have a question though. I'm always, so in this case, for example, cerebellum is not included in any of this, or is it? I didn't see it, but what do you think? If it's included at some point, do you think it will change anything or not? And cerebellum in this?
Well, it would change that you don't have the cerebellum, right? Like, but I don't think that. It is, yeah, I also do cerebellar research, so it's also important to consider the cerebellum, and maybe there are also the ICNIC maps available of the cerebellum, and then I would love to link it, because it would kind of be interesting to see how these atrophy maps of the cerebellum would relate to what you see in the OFC. I'm not sure what I would expect there, because, no, I'm not sure. Probably.
Yeah, we'll talk later.
Yeah.
Anyone else has a question? Yeah, sorry, there's a question here. How do you separate out effects of residual substances in the system versus straight or state connectivity differences in substance use disorder participants? Sorry, go on.
Here it was just cortical thickness, right? So I think that cortical thickness, we all agree on that it's not so much related to the state, but it's more trait. But yeah, you're right that of course there's this whole more neuropsychological component that at the moment we cannot speak about, right? So with the data that we have, we cannot draw any of those conclusions, and it's also not a causal framework. The only thing that we will do is also relating this to age effects to see that potentially the longer people have a substance use disorder, like how this will link to these changes. And there's a couple of other things that we will also test, but I guess this is also just a start for me to see this important comorbidity and, yeah, in a way, very, very interesting topic of study in relation to the brain and gene-environment interaction.
Okay, thank you.
Yeah.